Hello everyone and welcome to Module 9 where we're going to look at genetics, genomics, and epigenetics. In this module, I'm going to go ahead and give you a brief overview of terminology associated with this subject. Your learning objectives are going to be to review the components and steps involved in the central dogma of biology, differentiate between genetics, genomics, and epigenetics, and give examples of how environmental exposures are associated with epigenetic changes. So we all know that all multicellular organisms have a cell and a cell nucleus. This nucleus contains the DNA, the hereditary material. But this DNA does not float around randomly through the nucleus. It is neatly packed into chromosomes. People have 46 chromosomes, which means that you have 2 copies of each chromosome. So of the 46 chromosomes, you have 23 from your mother and 23 from your father. Except in the last couple of weeks in the news, there were reports of a baby born that had DNA from 3 different parents. The end of each chromosome is called a telomere, which protects the genetic data. Telomeres have been compared with the plastic tips on shoelaces because they keep chromosome ends from fraying and sticking to each other, which could destroy or scramble an organism's genetic information. Yet, each time a cell divides, the telomeres get shorter. When they get too short, the cell can no longer divide, it becomes inactive, and it dies. This shortening process is associated with aging, cancer, and at higher risk of death. In the Oliveira article, the authors reviewed the literature that examined the correlation between stress and telomere length, and they found that chronic social stress accompanies telomere shortening. This is important in population health, as many stressors have an environmental, behavioral, or socioeconomic component. Therefore, interventions at the policy level addressing issues such as poverty and gender equality and others are important, as it can alter the underlying physiology. So, all remember that each cell performs a specific function, and these functions are determined by the information contained in genetic material or the genome of the organism. There are 2 types of genetic materials in a cell, DNA and RNA. DNA is a molecule that contains all the biological information required to build and maintain a cell. Most DNA molecules consist of 2 polymer strands coiled around each other in a double helix shape, and each strand is made of 4 chemical bases: adenine, guanine, cytosine, and thymine. The base pairs that can be formed by these chemicals are limited. Adenine can only form a base pair with thymine, and guanine can only form a base pair with cytosine. So, if you know the sequence of bases on one strand of DNA, you also know the sequence of bases on the other strand of DNA. So when you hear people talk about DNA sequencing, they are referring to a technique that determines the order of the 4 bases, and it is the sequence of DNA that codes for the type of protein that is being made. RNA comes in a variety of shapes and types. It is usually single-stranded. It also has the same basis as DNA, except instead of thymine, it has uracil. The main job of the RNA is to transfer the genetic code needed for the creation of proteins. This process prevents the DNA from having to leave the nucleus and keeps both the DNA and the genetic code protected from damage. So without RNA, proteins could never be made. Proteins are large complex molecules that play many critical roles in the body. They do most of the work in the cells and are required for the structure, function, and regulation of the body tissues and organs. Proteins are made up of hundreds of thousands of smaller units called amino acids, which are attached to one another in long chains. There are 20 different types of amino acids that can be combined to make a protein. The sequence of amino acids determines each protein's unique 3-dimensional structure and its specific function. Proteins can be described according to their large range of functions in the body. For example, they can function as an antibody, an enzyme, a messenger, they can be a structural component, or be involved in transport and storage. So this leads us to the central dogma of biology. The flow of information from DNA to RNA to protein is one of the fundamental principles of molecular biology. The journey from gene to protein is complex and tightly controlled within each cell. It consists of 2 major steps: transcription, and translation. Together, transcription and translation are known as gene expression. So gene expression profiling identifies active and inactive genes in cell or tissue. Then this lets us know which genes may play a role in disease or condition. This is a very much a vital step in how organisms work. Next, let's look at genotype and phenotype. Your genotype is your complete heritable genetic identity. It is your unique genome that will be revealed by personal genome sequencing. However, the word genotype can also refer just to a particular gene or set of genes carried by an individual. For example, if you carry a mutation that is linked to diabetes, you may refer to your genotype just with respect to this mutation without consideration of all the other gene variants that you may carry. In contrast, your phenotype is a description of your actual physical characteristics. This includes straightforward visible characteristics like your height and eye color, but also your overall health, your disease history, and even your behavior and general disposition. However, not all phenotypes are a direct result of your genotype. Chances are that your personal disposition is the result of your life experiences with different factors rather than a mutation in a gene. Most phenotypes are influenced by both your genotype and by the unique circumstances in which you have lived your life, including everything that has ever happened to you. Therefore, we often refer to these two inputs as nature, the unique genome you carry, and nurture, the environment in which you have lived your life. I want to spend a little bit of time talking about genetic mutations. A gene mutation is a permanent alteration in the DNA sequence that makes up a gene, such that the sequence differs from what is found in most people. Mutations range in size. They can affect anywhere from a single DNA building block base pair to a large segment of a chromosome that includes multiple genes. Gene mutations can be classified in two major ways. Hereditary mutations are inherited from a parent and are present throughout a person's life in virtually every cell in the body. Acquired or somatic mutations occur at some time during a person's life and are present only in certain cells, not in every cell of the body. These changes can be caused by environmental factors such as ultraviolet radiation from the sun, or can occur if a mistake is made as DNA copies itself during cell division. Acquired mutations in somatic cells, these are cells other than the sperm and the egg cell, cannot be passed on to the next generation. An example of a somatic genetic mutation would be cancer tumors. Somatic mutations that happen in a single cell early in embryonic development can lead to a situation called mosaicism. These genetic changes are not present in a parent's egg or sperm cell or in the fertilized egg, but happen a bit later when the embryo includes several cells. As all the cells divide during growth and development, cells that arise from the cell with the altered gene will have the mutation, while other cells will not. Depending on the mutation and how many cells are affected, mosaicism may or may not cause health problems. Some conditions that are associated with mosaicism are Klinefelter syndrome, trisomy 18, and Turner syndrome. Genetic alterations that occur in more than 1% of the population are called polymorphism. They are common enough to be considered a normal variation in the DNA. Polymorphisms are responsible for many of the normal differences between people, such as eye color, hair color, and blood type. Although many polymorphisms have no negative effects on a person's health, some of these variations may influence the risk of developing certain disorders. A genetic predisposition, sometimes called genetic susceptibility, is an increased likelihood of developing a particular disease based on a person's genetic makeup. A genetic predisposition results from specific genetic variations that are often inherited from a parent. These genetic changes contribute to the development of a disease, but do not directly cause it. Some people with a predisposing genetic variation will never get the disease, while others will, even within the same family. For example, certain mutations in the BRCA1 or BRCA2 genes greatly increase a person's risk of developing breast cancer and ovarian cancer. Biomarkers, are any measurable characteristics of an organism that reflect a particular physiological state. In medicine, biomarkers are often compounds isolated from serum, urine, or other fluids that can be used as an indicator of the presence or severity of a particular disease state. Biomarkers can take many different forms, including particular proteins or peptides. For example, prostate-specific antigen as an indicator of increased risk for prostate cancer, antibodies for rheumatoid arthritis. Although physical traits or physiological metrics like height or blood pressure are biomarkers, the term is now typically shortened for molecular biomarker. Transcriptional profiling, DNA methylation studies, and kinase sequences are examples of some of the techniques that are used to look for biomarkers. Today, there are quite often news about specific biomarkers that have been identified for a specific condition. I will personally be working on a study where we are trying to find out whether there is a specific biomarker present in patients who develop chronic pain following injuries. This information can then be used to screen patients at high risk at the time of injury. So epigenetics focuses on processes that regulate how and when certain genes are turned on and turned off, while epigenomics pertains to analysis of epigenetic changes across many genes in a cell or entire organism. Epigenetic processes control normal growth and development, and this process is deregulated in diseases such as cancer. Diet and exposure to environmental chemicals throughout all stages of human development, among other factors, can cause epigenetic change that may turn on or turn off certain genes. Changes in genes that would normally protect against a disease, as a result, could make people more susceptible to developing that disease later in life. Epigenetic mechanisms are affected by several factors and processes, including development in utero and in childhood, environmental chemicals, drugs and pharmaceuticals, aging, and diet, a couple examples. Epidemiological studies have been carried out in which environmental exposures and lifestyle, such as diet, behavior, stress, physical activity, working habits, alcohol and tobacco consumption, meshed with genetic background and were associated with epigenetic changes. And you can read more about these in the Foley article. Alright, so how do we differentiate between genetics and genomics? Genetics is a term that refers to the study of genes and their role in inheritance. In other words, the way certain traits or conditions are passed down from one generation to another. Genetics involves scientific studies of genes and their effects. Genes carry the instructions for making proteins, which direct the activities of cells and functions of the body. Examples of genetic or inherited disorders include cystic fibrosis, Huntington's disease, and PKU. So for genetic testing, you really have to know which genes you are looking for. Genomics is a more recent term that describes the study of all of a person's genes, the entire genome, including interactions of those genes with each other and with a person's environment. Genomics includes the scientific study of complex diseases such as heart disease, asthma, diabetes, and cancer, because these diseases are typically caused by more a combination of genetic and environmental factors than by individual genes. Remember, nature versus nurture. So for your readings, you're going to be reading about stress and telomere length, aging and longevity, and epigenetics such as DNA methylation and stress. For your discussion for this module, we want you to find an article that explores the role epigenetics may play in altering the phenotype of your population of interest. If you cannot find anything related to your population of interest, you can choose a different population. So when you look for articles, I suggest you start off by searching epigenetics and the condition you are interested in. For my research, I would type in something like epigenetics and pain and environment. Some of these studies will be animal studies, and it is okay to review one of those. So when you read this article, do not get bogged down by the methods and results sections, which can be fairly complicated, but try to focus on the introduction and the discussion sections. After this, summarize the main conclusion, state what you thought was the most interesting aspect of this article, and then give an example of how this finding could be implemented at the population level. I have also provided you with a link to some websites that have additional information about genetics, genomics, and epigenetics. So I hope you enjoyed this lecture.